Hello, I'd like to welcome everyone to today's webinar, sponsored by the UCLA Division of Physical Sciences, entitled The Science of Our Lives, Research That Matters During a Pandemic. My name is Miguel García-Garibay. I am the Dean of the Division of Physical Sciences in the College of Letters and Sciences. I would love to be able to welcome all of you today at the beautiful UCLA campus here in Westwood. However, today we cannot do that, and however, we can join all of us today by this medium. So in the next hour, we will hear from 3 faculty members from departments in the Division of Physical Sciences who will share with us their expertise on issues related to the ongoing coronavirus pandemic. After 3 presentations, we will have time to select a few questions from the audience, and we will hear the answers from our speakers. So we will get right to it. So our first speaker is Professor William Gelbart. Bill is a physical chemist, a member of the Department of Chemistry and Biochemistry, who along with his collaborators has been studying viral particles for a few years. So thank you, Bill, for sharing a little bit of your work and your expert views with us. So whenever Bill is ready.
I'm ready.
Wonderful. Welcome, Bill. The floor is yours.
Okay, thank you, Miguel. And I'm very glad to be here and have the chance to tell you a little about the work we're doing, how and why we're doing it. Indeed, I want to tell you about a new idea that we're pursuing for a vaccine, and in particular for COVID-19. We've been working for years on viruses. We try to work on the simplest ones we can. Coming from a physical background, moving into molecular biology, it was natural to ask, what are the simplest viruses and can we figure them out first before we go to the more complicated ones? So I want to acknowledge first my research group. I've underlined just a few names here, and that's because for safety reasons, we're only able to have a few people in the lab. And the underlined people are the people whose work I'll be telling you about. I want to mention also, most importantly, a collaborator in the School of Medicine, a virologist, immunologist, Professor Otto Yang, who's been extremely helpful. The story I'm going to tell you briefly is one of 3 viruses and a vaccine. Basically, I want to explain why we bother with 3 viruses in order to make a vaccine. Clearly, the vaccine is against SARS-2. It's the pandemic we're reeling under, but there is a sense in which all viruses are the same and in which all vaccines are the same. I'm going to be explaining why we have been able to bring to bear on our work what we've learned about a very simple plant virus. It's called CCMV. The name isn't important. A very simple insect virus, Nodamura, and how we use our knowledge of these 2 viruses to design a new vaccine for COVID-19. I'll start with the punchline showing you our vaccine idea. This is the slide I'll be showing you at the end of the presentation. But I want you to have a feeling from the outset for how we use components of these much simpler viruses in order to do the best job of developing a new and hopefully effective vaccine against COVID-19. So what you're seeing here are cartoons of the pair of vaccine particles that we're busy at work making. You'll see that the one on the left, the one we call a B particle, has familiar looking spikes. You've seen lots of pictures of the coronavirus, and you've even heard in particular about its arguably most important protein for vaccine reasons, and that's the spike protein that gives the virus its name. It suggests a corona. So the blue particle on the right is a CCMV capsid. What's a capsid? It's just a protein shell that protects the genome of a virus. The particle on the left, which we call a T particle, is the very same capsid formed from the capsid protein of the plant virus CCMV. But the working part of the virus here, instead of being the spike proteins of the coronavirus, it's an RNA molecule. We call it a replicon because it's a self-replicating molecule. I'll explain what I mean by that. And it has sequence that comes from several proteins of the coronavirus. Basically, it's the sequence in the RNA in this particle that's going to elicit a killer T cell response from the immune system. It's the intact proteins, the spike proteins here presented on the outside of the virus particle and the B particle that will be eliciting an antibody response. So what I'll be telling you about is how and why we developed the strategy and why we think it will work. These are hybrid particles. They're not viruses because they're not infectious, but they're virus-like particles, and they're made from purified components that we get from these three viruses. We take the capsid protein of CCMV, we take the RNA from the insect virus, and we take the spike protein from the coronavirus. First, why are we bothering with the plant virus? This plant virus is special because it was the first spherical virus to be made from scratch. The purified capsid protein of the virus, the purified RNA genome of the virus, mixed together under the right solutions, the conditions, give perfectly formed, fully infectious virus particles. They're perfectly symmetric, as you see. And it's clear that this is a way to understand a virus. Take it apart and put it back together again. If you can do that with a radio or television set or a computer, you understand a lot about it. So that's been our strategy all along, to go to the simplest viruses, take them apart, put them back together again if we can. So we're going to be using the capsid protein from the plant virus because we found that we can mix it with any RNA. It doesn't have to be the RNA from the plant virus. By the way, when I say plant virus, it's a virus. It's not a plant, but it infects plants, okay? Why the insect virus? Again, this is a virus that infects insects. Again, a virus like the plant virus, like coronavirus. Its genome, exceptionally, is RNA. And what's special about this virus, and the reason we've been studying it for several years now, is because not its capsid protein, but one of its few genes is important to us, namely a gene that codes for a special enzyme, a protein called RNA replicase. It's called replicase because it replicates RNA. When this RNA molecule is translated into protein, you get RNA replicase protein. So what we do is to use this gene to make self-amplifying forms of important therapeutic genes or vaccine genes. So we simply add the vaccine gene, the RNA sequence coding for viral antigens. We add it to the RNA replicase gene, and now we have a molecule that's self-amplifying in the sense that when it's translated into protein, it forms these two proteins, one of which starts amplifying it up to 1 million-fold. That means you generate a lot of these two proteins when this molecule that's being amplified is translated into protein. And if a vaccine protein is good to have exposed to the immune system, you want a lot of it. So this is the pair of new ideas using the capsid protein from the plant virus and the RNA replicase gene from the insect virus. Now we have to talk a moment about the target virus. You've seen cartoons like this. You know that the coronavirus has lots of proteins. It's the most complicated RNA genome virus. Arguably, the protein that's been featured, and for good reason, most is the spike protein. It's on the outside of the virus. It's what presents the virus to the immune system. I remind you that vaccines are just antigens, things that stimulate a response from the immune system. They're generally administered in protein form. You can just take the spike protein and use it as a vaccine, inject it intravenously, or you can complex it with a delivery particle to protect it, target it to where you want it to go. But you can also deliver vaccines in gene form. They're simply RNA or DNA sequences that code for the viral antigen. Our idea, as you know now, is to present the antigen in both protein form and in RNA form. But what's new in both cases is that we're presenting the protein on a scaffold that's perfectly regular, and it's known that regular presentation of many copies of an intact protein antigen elicits a stronger B cell or antibody response. Similarly, if it's good to introduce a vaccine in RNA form, why not introduce it in self-amplifying RNA form? And that's what we do using the replicon from the insect virus. So it's this pair of vaccine particles that we deliver in tunable proportions so that we can control, hopefully, the relative strength of the antibody immune response from B cells and the killer T cell response from T cells. My time is up, and I want to come back to the research group because obviously they're at the heart of all of this. I mentioned before I was underlining the names of the few people, and I know how they feel. They're the ones who are lucky enough to be back in the lab. We had to close our lab like most labs 2 months ago, but we were able to get started because up to 3 people could be made exempt to work on this from the shutdown on campus. Everybody else in the group at regular meetings, a few times a week, has been supporting their efforts with ideas and all sorts of other resources. I mentioned already our collaborator in the School of Medicine. What I didn't mention is that we were able to get started because the UCLA AIDS Institute thought our idea was worth supporting. And on that note, I want to say that there's no better place I could be than UCLA to try an idea like this, because long before the urgency of fighting a pandemic, going back decades at UCLA, I found if I or someone else has a good idea, they get good support intellectually and in resources from lots of great collaborators and the whole university community. I'm looking forward to discussion and questions and answers.
Thank you very much, Bill. That was fascinating. I think that reminds us of the mythological chimeras, but this one is a combination of a plant, an insect, and a mammalian virus. It's just fascinating. Thank you.
Thank you.
So our next speaker is Professor Rick Schoenberg from the Department of Statistics. Rick applies sophisticated statistical methods to analyze a wide range of natural phenomena, from earthquakes to wildfires to the spread of infectious diseases. So welcome, Rick. We're looking forward to your presentation.
All right, thank you very much. Thanks for inviting me to speak. And let's see, again, hopefully you can see my screen here, okay? And I'd like to talk about the models currently being used to forecast the spread of COVID-19 and the next generation of models that my colleagues and I are working on that seem to be even better. But first, let me just tell you, I should tell you a little bit about how my background and about how I got into this. I'm a statistician, And I did my PhD on models for forecasting earthquakes, or more specifically on methods for evaluating how well those models fit. And it turns out the best fitting models for earthquakes are called Hawkes models, or slight variants of Hawkes models. And about 9 years ago, some colleagues and I decided to try to tailor these Hawkes models so they could forecast other things like disease epidemics and invasive species and other things. So it turns out they work really well there too. But the main models used to forecast COVID-19 today are SIR models, and where the population is categorized into these different compartments, the susceptible, the exposed, the infected, and the recovered or deceased. And there's some rate at which people are going from one compartment to another. And there are many little variations of this, a bunch of equations that are supposed to govern this. Beta is the transmission rate constant here, and gamma is called the recovery rate, and their ratio, R0, is called the reproduction number. And traditionally, all those things are fixed, but you can model these things as changing over time as the epidemic progresses. So R0, R0 is the reproduction number at the beginning of the outbreak, and as it changes over time, we call it R. And R measures how many people are infected, like how many people each infected person is expected to spread it to on average. So if R is greater than 1, the disease is multiplying quickly, it's explosive, whereas if R is less than 1, then it's dying out. And this plot, which was made by the LA County Department of Health Services Task Force that I'm a member of, and it shows how estimates of R have changed as a result of distancing measures. And the task force is using SIR models and trying to forecast things like the number of ventilators that are going to be needed, the number of patients that are going to be admitted to local hospitals and things like that. And I have to say, I've been pretty impressed with how well the model's done so far, particularly around April 15th. I don't know if you could see, let's see on the next plot. Around April 15th, there were these 3 points here. Hopefully, you can see that. If I can make this bigger. Maybe that's better. There were a few points here. This was a point in time. This, by the way, is about when I got the disease, and it's very weird to be studying a disease while you are suffering from it, I have to say. But around the middle of April, there was this point where we had about 3 really high points in a row, and no one really was sure whether it was going to shoot up again or stay flat or go back down. Here we have the number of new cases on the y-axis just in LA County. And, yeah, it was a very uncertain time. We really didn't know what to expect. And the model said that it was going to stay flat, and it mostly did. So that was kind of impressive. There are, in general, some impressive things about SIR models. They are very widely used, and that was very important to the task force. They're very standard. They make sense. They're reasonably accurate for short-term forecasts, but they have problems. The equations governing them can be wrong in practice. They seem to depend very critically on things like the overall number of people who've gotten the disease, the asymptomatic,
Hello, Rick. We're getting some feedback from some of our viewers that if you could put it in presentation mode, some people still are not able to see.
Oh, great. Thanks for telling me. Sorry about that. Okay, where was I? Yeah, there are some problems with SIR, like the equations can be a little wrong. We call that misspecification. It depends critically on some things that are really hard to estimate, like, what's the incubation time for the disease, how many people are asymptomatic, things like that. It's famously, there are certain cases where it's had low accuracy, especially for long-term forecasting. There was, for the first SARS outbreak in China, for example, in 2003, SIR models estimated that there were going to be between 30,000 and 10 million cases of SARS in the first 4 months. And ultimately, there were like 700 something reported. So, it can have low accuracy, especially for long-term forecasting, and it can even be poorly calibrated, meaning like in Italy for COVID-19, it's the forecasts of deaths in Italy were about 14% too low on average. So even on average, it can be wrong. So there's some reason to hope to do better than SIR models, and I think Hawkes models are the answer. The idea behind Hawkes models is that there's some background rate, mu, which is the rate at which infected people are coming into your city, and then those infected people have some productivity. We call this cake productivity, and so they're giving it to other people at some rate, and those people are like their aftershocks, and those aftershocks have aftershocks, and so on. And so my colleagues and I proposed this, what's called the recursive model, which is where this productivity changes over time as a function of the overall rate. But anyway, there are certain variants of the Hawkes process that are currently being studied. And this is the main slide I wanted to prepare for this talk. So I looked up all the papers I could find that compared the errors, the forecasting errors of the SIR model with Hawkes models and their variants, like the recursive model. And overall, you could see some of these authors, by the way, are UCLA people, but the point is the Hawkes and recursive models in almost every case fit better, have smaller root mean squared errors, so smaller typical sizes of the forecasting errors by about 20 to 30% typically. And so it seems to be a very promising technique to get even more accurate modeling. You know, SIR does fine, but these models seem to do better. And I've been looking into other ways to assess these models. This is data on pertussis in Nevada over a long period of time. And we have SIR on the left, SEIR models on the left, and we're the recursive model on the right here. And these are called super thinned residuals, where the point is you're supposed to look for uniformity in these residuals. And if you see any gaps like there are here or clusters, that's indicating where a model might be improved. Whereas with the recursive model, it looks really good. And so we're working on residual methods like this so we could see how to improve the models. And that's basically it for my talk. I mean, the SIR models seem fine, but I think Hawkes and recursive models are going to be the next big thing in forecasting the spread of these contagious diseases. They're already being used by the state of New Jersey's health department for their forecasts of COVID-19, but most other places are still using SIR models or their variants. We need more evaluation techniques, and residual plots may suggest areas for improvement of these kind of models. All right, thank you very much.
Thank you very much, Rick. That's fascinating. I think we can certainly take advantage of those models for many applications. So thank you so much. Our next speaker is Professor Suzanne Paulson from the Department of Atmospheric and Oceanic Sciences. Suzanne is also the chair of the department, and she studies all aspects of air quality, which in cities like ours is of course determined by human activity. As you can imagine, the stay-at-home order makes it possible to look at changes in that would have not been possible otherwise. So this is like a very unusual experiment, and Suzanne is going to tell us about that. Thank you, Suzanne, for joining us today.
Thank you, Miguel, for that introduction. Why are these not advancing? So for some reason things are not advancing the way I expect. All right. So I want to first tell you that air quality in Southern California has improved tremendously over these past many decades. It's at least 75% cleaner than it was at its worst. And this is a testament to advancements that result from science, from engineering, from policies, and from political will. And some of that science comes from my department. However, the area still has levels of ozone and tiny particles smaller than 2.5 microns, or PM 2.5, that are too high. So during the stay-at-home order, we saw some really remarkable headlines. Things like, LA's air, unusually terrible, is the best in the world. And this is consistent with reports we were getting out of Asia and out of Europe. And on the right, there's satellite images or satellite data products of nitrogen dioxide pollution over Europe from March, April 2019 and 2020, and it's down by around 50%. So we might expect that air quality got much better in Los Angeles. So in Los Angeles, during the initial part of the closure, cars were down about 45% on the freeways, more on surface streets. Trucks were down by 25%. Activity in the ports was down by about 10%. And then we can look at the actual PM 2.5 or fine particle concentrations. And in this plot, we have on the x-axis kind of the air quality index, its concentrations, but normalized to health levels that are pegged to health outcomes. And the gray line is the average of the last 5 years. And the blue shaded area is the range of all the values in the last 5 years for the period of January until yesterday. And the blue line is what's happened this year. And you see that in March and April, the concentrations were really low. Actually, some of those values are lower than any other values we've seen in the last 5 years. There, however, is the stay-at-home order. So after some of these low values started to show up. And I would like to say that it was due to UCLA. That's the time when UCLA went online, and UCLA has huge impacts in the area, but I don't think we can really attribute the clean air to UCLA's activities. This is what else was going on. This is the rain. So we had a very dry January and February, and then in March we had a bit of a rain miracle. And you can see that the low levels are actually very closely related to the rain events. And you're not imagining this when you think the air is really gorgeous and clear after a rain event. The rain very effectively captures all the particles as it falls. So rain was a major part of what was going on there. We can look at the sources of pollution of fine particles in Southern California. And this is data, actually all the data in this presentation was collected by the South Coast Air Quality Management District. So there are a bunch of really smart people who work really hard and try and figure out where the pollution is coming from. It's not trivial, there's certainly some uncertainties, but basically on-road vehicles, trucks and cars are only responsible for about 16% of the little particles. The largest category in their inventory is miscellaneous processes. What is that? Well, the largest part of that is cooking, and then there's residential fuel combustion. There's also a component from the roads, paved road dust. But taken together, the changes in activity, mostly reductions in traffic, should have reduced the emissions of PM 2.5 by around 10%. So maybe a little bit of what we saw was the result of changes in very dramatic changes in economic activity. So how about ozone? So ozone forms in the atmosphere from chemical reactions of nitrogen oxides, or NOx, and reactive organic gases with energy provided by sunlight. So because they need sunlight to drive the reactions, ozone concentrations are typically quite low in the winter, and then as spring comes along, they get higher. So we see during the first part of the closure that, and also the rainy period, concentrations were really very low. But then in May, they got high. In fact, they got really high. This is one of the highest values we've seen in May in many years. So if anything, the ozone actually got worse. So ozone comes from nitrogen oxides. Nitrogen oxides are basically entirely produced by combustion processes. The largest share is on-road motor vehicles. But it turns out that almost all of that is trucks. Only about 12% is passenger cars. And this is really a tribute to the tremendous progress that California has made in cleaning up the vehicle fleet. 20 years ago, 30 years ago, the cars were a much, much larger share and even the majority of pollution in general, but we have regulatory authority to address passenger autos, and we have used that very, very effectively. We have less authority over things like trucks. They are involved in interstate transport, and the state doesn't have control over that. The largest part of other mobile sources actually is ships, which I also don't have much to say over. When we looked at the reactive organic gases, we see that motor vehicles are a part of it, but the largest category is actually solvent evaporation. And the largest contributor to that is consumer products, so stuff that each one of us does. And a large part of that is vapors from cleaning agents and disinfectants, which I'm sure you can imagine, the use of those has gone up a lot. And some of the molecules that are coming out of those products are quite reactive, more reactive than the average. So that may actually have increased reactivity of some of the chemistry and contributed to some of the high ozone levels, although there are probably some other things going on as well. So in summary, the air quality got much better in much of the world during the stay-at-home and lockdowns, and it really did. But in Los Angeles, the improvements were slighted fast, and ozone seems to have even gotten worse. And this is paradoxically a good thing, because it is a testament to the massive progress that we've made in cleaning up the air. So the vehicle fleet that we have on the roads right now is quite clean. The fuel that we buy reduces the emissions in all the cars that are on the road, so the fuel is quite clean, and as a result, the vehicles are no longer the dominant source of pollution in the Los Angeles area. So I did say in my title I would talk a little bit about filtration of very small particles. The virus that causes COVID-19 is very small. It's about 0.1 microns across. And most of the particles that are expelled by breathing and talking are around 1 micron, also very tiny. Some of those, if someone is infected, contain the virus. Coughing and sneezing can produce droplets that are, some droplets that are larger and are visible to the eye up to about 75 microns. So human hair is about 60 or 70 microns in diameter. We can see that obviously. Once we get down to 30 or 40 microns, we can't see it anymore. All these particles are much smaller than that. And all of these particles are much too small to be quickly removed from the air by gravity. So even the largest ones only fall at a rate of around ½ an inch per second, and the other ones, the 1 micron particles, stay in the atmosphere for an average of about a week. So they take a long time to be removed unless they encounter a surface, and it turns out that they're quite sticky. So they tend to, if they encounter a surface, they tend to stick to it. So N95 masks and HEPA filters and other filters designed for very small particles are typically rated for their ability to remove 0.3 micron particles. And that's because that size of particle is the size that is hardest to remove. And you would think that particles smaller than that would go right through, or go right through more effectively than 0.3 micron particles. But actually, the opposite is true. Smaller particles are filtered more efficiently than these 0.3 micron particles. All of these particles are not being removed the way that we think about filtration that we can see. So it's not that there are somehow holes that are smaller than these sizes that are stopping the particles from going through. If there were, we couldn't get enough air through them to do what we need to do. Like breathe, for example, in the case of a mask. And this is an electron micrograph showing submicron particles not going through holes that are much bigger and just kind of collecting around the edges and forming these little domes. And this is because they literally diffuse out of the airflow and stick to whatever surface they encounter. As a result, cloth masks that you would think wouldn't do much for these tiny particles actually can remove some of the particles. The cloth is not carefully designed for optimal particle removal and for the air to go through, so it's not really possible to make a cloth mask you could breathe through and that would collect all the particles, but they do significantly reduce the amount of droplets and particles that you exhale and as well as inhale. So they protect you and they protect everyone else more effectively than you might think. So thank you very much.
Thank you very much, Suzanne. That was fantastic. It's really interesting, you know, how complex our air quality can be and all the factors that contribute. So I want to invite the rest of the speakers to join us now. We've heard just the incredible presentations about using viruses to fight virus, mathematical models that help us prepare better on how to address the different issues and challenges that viruses pose, and how changes in human activity affect our environment, what's been done to do it, and actually that those viruses become part of our environment also, at least for some time. So I want to thank all of our speakers for really helping us you know, understand some aspects of the pandemic that might not be that obvious. And I think now we can select some questions from the audience and that we can share with you. So let me see. I'm gonna check my boxes here. Let me see. I have the first question. So the first question is for Bill. Bill, how do you take your vaccine from the academic lab to the general public? What's the timeline for your vaccine that is in clinic? Is it in clinical trials?
Okay, no, we're nowhere near that. We're at the stage where we're making the vaccine particles, these two kinds of particles with RNA and with intact protein antigen being presented. So we have two people working on that, a third person working on tests of those particles once they're synthesized. But these will be tests not in clinical trials. First, we work with immune cells, T cells, B cells, antigen presenting cells in the lab, and we ask, how are they activated by our vaccine particles? It's only when we get a positive response there that we think about either animal models or going directly to phase 1 clinical trials. And as you've all heard a lot about in the news, you're following closely, I'm surprised the first question wasn't simply, it was part of the question you posed, when will the vaccine be ready? And if yours isn't ready, when will another one be ready? And I think you appreciate the fact when you listen to experts seriously, nobody can predict that. So there are hundreds of labs in the world working on different ideas, and one of them will work first. Let's hope it's soon. But there's no way to predict the timeline. I can just tell you that we're in the very early stages, working as hard as we can.
Thank you, Bill. The second question is for Rick. Is your research and modeling able to predict when we might see a second wave of COVID-19 infections?
Ooh, not really. We don't know. It's in past epidemics, it's rarely been anything like this one. So honestly, no. Sorry, I have no idea. I mean, I've read other people's work, but it seems very speculative about predicting a sort of second wave coming in the fall. It's based on a lot of assumptions that seem really tenuous to me. I'm not saying it's wrong, but it seems like guesswork a little bit to me. And the Hawks model really does not do any good at forecasting when a second wave might come, unfortunately.
It sounds like it must depend a lot on human activity, right? How well we take care of ourselves and factors that are not easy to know without knowing the facts.
I would think so, yeah.
Yeah. Okay, let's see. So Cindy says, I attended UCLA in 1977. The smoke was so bad, it took months for me to realize that the mountains beyond Pasadena existed. The sky was orange almost all the time. So for Suzanne, why are 0.3 microns the hardest to remove?
Oh, that is a fascinating question. But basically, particles are removed by settling due to gravity. It's slow, but it does work. And they're removed by diffusing out of whatever airflow is going by. they just kind of move by Brownian motion out of the airflow and stick to surfaces. And in filters, actually, we can have some of the fibers charged, so we can sort of add electrostatic attraction to that mechanism. And then they also, when they're going around a bend, if they have a certain amount of mass, they'll go straight and they'll stick to whatever they impact. So particles that are larger than 0.3 microns have reasonably large gravitational removal, and particles that are smaller than that diffuse pretty effectively. And it's the 0.3, 0.4 microns that don't do either of those things very well that stay in the air for the longest time. And it also turns out that they scatter visible light the most effectively. So when the air looks hazy in LA, unless if it's not raining, then that's what it is those little particles scattering light.
Thank you. So let me see, the next question is for Bill. Will this technology that combines different viral components to create a vaccine be applicable to other viruses that do not have vaccines yet, such as MERS or AIDS?
Great question. One way to answer it is to say that, in fact, it is a very general platform. And the reason we were able to get such a quick start, we weren't working on coronavirus months ago. We were working on these other simpler RNA viruses, but we were already working for years on HIV vaccines, which is how we knew about the UCLA AIDS Institute. Irvin Chen, the director, knew about our work. We also were working on cancer vaccines. So the platform is a perfectly general one. You've heard about cancer immunotherapy. We're talking now about viral immunotherapy. So we have our way of packaging, protecting, and targeting RNA in these particles that we can make from scratch in the lab, and we have our ways of presenting protein antigen. So it's a general platform. It's applied to coronavirus now because of the urgency of the situation. But we hope we'll be in a position where we have the luxury of going back to working on an HIV vaccine and going back to working on cancer vaccines because we or someone else will have made the vaccine work in this case.
Wow, that sounds really interesting and very, very exciting. Thank you, Bill. Let me see. The next question is for Rick. To what extent has the Hox model been applied to predict the spread of other infectious diseases besides COVID? And is it more effective in cases as well?
I'm sorry, I missed the last part.
The last part. And is it more effective in those cases as well, if it has been applied in other infectious diseases?
Yeah, in almost every case, it's been a more successful predictor than SIR models. So we've used the Hox model to successfully forecast Ebola in West Africa, in 3 different countries in West Africa. It was very, very successful, had much lower error rates there than SIR models, which had already been published to forecast Ebola in those places. And then also in diseases in the United States, I mentioned pertussis in Nevada. We looked at Rocky Mountain spotted fever cases in California going back several decades, and Hox models and their variants were very successful at forecasting those as well. And so it really looks like, and mumps also, mumps in Pennsylvania. My colleagues and I have a paper on that. It looks like for various different contagious diseases, these Hox and recursive models seem to fit really well.
Thank you. In fact, you answered already a follow-up question. So the person who asked this question wanted also to see if this would work in places like Tokyo, and you're telling us that is really robust in every, basically, geography. So let me see.
I think so.
Thank you. Let's see. The next question is for Suzanne. As we are starting our return to work in big buildings, are you knowledgeable about what big buildings have in the way of filtering particles?
That's an interesting question as well. So, so most big buildings have mandated air change rates of I don't remember exactly, but at least couple or a few per hour, which means that the air is going through some sort of mechanical air handling system. And the particular filtration that's in a given building varies, but so if they have HEPA filters, then the air should actually be pretty effectively cleaned out. But even if they don't have any specific filtration, then the concentrations of particles will go down at least by 50% every time they go through the mechanical air handling system. So, in general, having the air conditioning on is not great for energy consumption, and certainly climate is a huge threat. But having the air moving around through a mechanical air system, air conditioning system, whatever, does definitely appreciably reduce concentrations of particles, including the viruses.
Thank you. Let's see. So the next question is for Bill.
You're surprised each time, but obviously we're going in a cycle. People feeling the question.
Yes, I am getting them in a cycle.
You're doing a good job of pretending to be surprised each time.
So why not replicate the entire virus minus the internal RNA? Wouldn't this be more effective?
The virus can't be replicated without the RNA. All RNA viruses need to code for the special enzyme RNA replicase, which they will not find in a host cell. Only viruses replicate RNA. If we're replicating RNA in our bodies now, it's because we're infected with an RNA virus. So you can deactivate a virus many ways, and the whole point is it can't be replicated. The surest way to do it is to get rid of the RNA. Mm-hmm. But then you're left with deactivated virus vaccines, where you have all the spike proteins on the outside and so on. You are eliciting an immune response, but you're not getting a full immune response because you're not synthesizing in host cells the viral protein antigens from the viral genome. Mm-hmm. That's why we think it's so important to be doing both in the same vaccine.
Thank you, Bill. I would not look surprised, but the next question is for Rick. Who outside of UCLA and local health departments are you collaborating with on modeling?
Oh, well, I'm working with several people at UCLA, and I'm collaborating now with the LA County Department of Health Services, who are using Berry consultants also. So I'm working with them. They've been outsourced by the county. But also I've been working with some people from other schools that we don't like to name, but in Northern California as well. But really there's a worldwide effort to try to get these kind of forecasting models for epidemics tested in a more organized way against each other, so that we can really improve the models and see which ones are working better in what cases. And it's sort of similar to, well, it's coming along. So I've been working loosely with some people from World Health Organization to try to get that set up. We're basing it on actually another thing that UCLA is involved in, which is forecasting of earthquakes. There are these experiments set up where you can input a model for forecasting earthquakes, and there's sort of a testing center where they're all competing against each other. They have more than 100 models. And we want something similar set up for disease epidemics. So the World Health Organization and others are working on that.
Mm-hmm. Thank you, Rick. So for Suzanne, of course. It is expected that global warming will increase the incidences of microbial bacterial outbreaks. Do you think that this would also affect the viral activity? Do you find a correlation between pollution and novel viral activity?
Well, I'm not really a virologist or bacteriologist, so I can't really speak to how they respond, particularly to temperature. But there have been some early studies indicating that people who live in areas where they've been exposed to higher levels of air pollution have had worse outcomes from the coronavirus. Those results are pretty preliminary, and it does, to the extent that they've come out, seem to be more associated with long-term exposure, so that the short-term drops during the coronavirus, you know, maybe mitigate that and maybe don't.
Thank you. And so now, perhaps Bill can let some insights into this question, into viral behavior. What do you know, Bill, about, you've been growing viruses? I know the physical chemistry of viral growth. So do they replicate better at higher temperatures, perhaps?
When viruses aren't in their host cell, they're in trouble. So temperature is a bad thing for them. But of course, they live in our bodies, and our body temperature is in the high 90s. That's a hot day, becoming more and more common with climate change. But I would say the effect of a few degrees in temperature on viruses when they're outside their cells that host them, it's a minor effect. Mm-hmm. So I would think the temperature effect is more a purely physical one. And so it would come back to Suzanne's court. To what extent do aerosols and their stability depend on temperature? Mm-hmm. Because the virus is at 99 Fahrenheit, 100 Fahrenheit, it's all the same to them. That's their temperature in a host cell, but they have all the help of the host cell. So.
Mm-hmm. Right. So a high fever is not going to be particularly helpful in that regard. Let me see. Now I have a question for Rick. What does the R behavior so far tell us about the required level of prevalence of individual immunity to establish herd immunity? Do the SEIR of the hogs models accommodate the effects of immunity developed by recent recovered patients?
Wow, okay, that's a tough question. And it's a little bit outside of my wheelhouse. I mean, I will say I showed the simplest version of the SIR models in my slides, and there's all kinds of variations. And the way that these models are fit nowadays is you let the R change over time. And what the best we can do is estimate it, how it's changing over time. And we can clearly see that the effects of physical distancing reduced R in LA County, for example, from something between 3 and 4, somewhere around 3.5 to right around 1 now. So it's pretty clear, and we see this all over the world, that physical distancing has this kind of an effect. But the technical, parsing out how much does this matter and that matter, that's beyond my knowledge or expertise. And then also your other question about herd immunity, I mean, I've read things about it, but it really, my research doesn't speak to it, so I probably shouldn't answer. I don't know really much about herd immunity.
Thank you, Rick. So the final question is really for all panelists, all of you. What are the most hopeful, what are you most hopeful about when it comes to how your area of science can help us through the pandemic? And again, maybe we'll go in the same order. We'll start with Bill.
Yeah, obviously it's with vaccines and treatment. Something I didn't mention before is that vaccines aren't helpful only as prophylactic or preventive measures, but also for treatment, for therapeutic purposes. You can use a vaccine to attack virus in a person who's suffering from COVID-19 disease. So I think here the answer is clear. We need vaccines for prevention and for treatment. And what I'm hopeful about is that one thing that the pandemic has engendered is a lot more cooperation among scientists, less competition. I think we see a new road to collaborating. Funding agencies are changing how they fund science, thinking about getting the right people together quickly and so on. Similar thing happened with the Manhattan Project during World War II, and science has been practiced very differently as a result of people coming together to understand real threats to society. So we need vaccines and treatment. And as we were talking about before, if we understand more about coronavirus, we understand more about HIV and ultimately even about cancer vaccines and so on.
Thank you, Bill. Rick?
I mean, this virus has a pretty long incubation time. So for me, I mean, one of the real values of having good forecasting models is that hopefully we can, as quickly as possible, determine when we're going to need to reimpose or have stricter stay-at-home orders or physical distancing policies, and hopefully we can base that on data very quickly as they come in, so we could see if another spike is happening and if that's going to mean that we have to have stricter physical distancing policies.
Thank you, Rick. Suzanne?
So I think that the coronavirus epidemic, pandemic has gotten a lot of people to think more about science, which I hope in the long run is good for science. And I hope that, and I think a lot of my colleagues are doing their best to put a good foot forward and communicate well about science. I think that's super important in times like this. And I'm hoping that the disruption will result in positive changes in people's habits and choices that will end up improving air quality and also our impact on climate change.
Just a note about, yes, science is overwhelmingly the key to solving these problems. We have to be careful not to have unrealistic expectations for science. The fact that some viral diseases don't have vaccines after decades of work should make it clear that you're lucky when you find a vaccine. It's not a fault of science when you don't have a vaccine within months, years, or decades. So in the end, we make progress, but not on a predictable, sure progress, but not on a timetable. These are tough problems, fundamental processes and interdisciplinary problems that we are working on better all the time, but they don't get solved overnight, and we shouldn't have unrealistic expectations about that.
Yes, well, thank you to the three of you. I think the points that you're making at the end are extremely important, right? This pandemic has exposed the fact that there's so much more that we need to know, that there is so much fundamental knowledge about something as simple as a virus and how something as simple as a virus can affect our entire society, which reflects the fact that we need physical scientists like ourselves working with life scientists and social scientists and humanists, and really all of us working addressing these challenges. I think our audience can see how excited we are about our work, about what we do. We talked a little bit about how one of the main things that we do is teach our students to be prepared to tackle these challenges in the present and in the future. I want to also be really acknowledge the fact that many people in our society are really having a very, very difficult time right now, that this pandemic is affecting all of us, and some people in our society are much more vulnerable. And I think UCLA is really being a leading force and trying to bring social equity in every aspect. All of us have a really deep sense of community. I think that's what makes Bruins very, very special. And I think our listeners, our visitors today should be assured that all of us take our responsibility to not only to produce the basic science that will help improve our standards of living and address challenges, but we also really, our primary goal is to really train and mentor our undergraduates, help them be the best they can be. I want to finish by saying that this was the first of what I hope will be many webinars when we will be learning about what our colleagues in the physical sciences are working on, whether it is aimed at the coronavirus pandemic itself or other aspects of science that we really would like to know more about. Things like quantum computing, for example. What is the future going to be like in many aspects of technology? So I want to just thank our speakers, Professor William Gelbart from the Department of Chemistry and Biochemistry, Rick Schoenberg from the Department of Statistics, and Suzanne Paulson from the Department of Atmospheric and Oceanic Sciences. And behind the screen, there's a team of people at UCLA that are helping us putting this together. I help our team, I thank our team for their help. And I thank all of you for your attendance. We hope to see you soon. Thank you for joining us, and goodbye.
Thank you very much, and great job, Miguel.
Thank you.
Thank you. Goodbye.
Thank you, everyone.